Okay, question number 1. The most abundant molecules in the plasma membrane are, okay, and we have glycolipids, phospholipids, proteins, etc. So let's take a look at the figure. Okay, and right here we have a figure of plasma membrane. And as you can see here, most of it is golden, right? So then golden, what is golden? This right here, you can see the brackets right there. Those are going to be phospholipids, right? So therefore, the most abundant component is going to be phospholipids. So let's go back to the question. And right there, you can see phospholipids. Next question. The plasma membrane of cells lining the small intestine has blank, which are specialized for absorption of nutrients. And we have microvilli, flagella, glycocalyx. So let's take a look at the figure. I'm going to stop for 5 seconds so you can think about your answer. And now in the figure, we have the microvilli, which increases the cell surface area for absorption. Absorption, obviously, of nutrients, right? And that happens at the level of the intestines. Going back to the question, that's what we have right here. Microvilli, and we're going to have that in the small intestines. Next question. Blank is the process in which blank forces water and small solutes, such as salt, through narrow clefts between capillary cells. And we have filtration, osmosis, active transport. Okay, let's take a look at the figure. Okay, and we have filtration. Filtration happens due to the hydrostatic pressure, right? That's the force that is going to push basically solutes, as you can see right here, outside the capillaries. This is a capillary, right? Going back to the question, right there, filtration and hydrostatic pressure. That should be the answer. Next question. Sodium and glucose are transported together from the intestinal lumen into an intestinal cell. The carrier protein is a blank, and the process is called blank. And the options are going to be symport, antiport, uniport. So let's take a look at the figure. I'll give you 5 seconds so you can think about the answer. And now in the figure, we have types of carriers, right? We have uniport, which is going to transport only one thing at a time. And we're going to have symport, 2 or more solids in the same direction. Or antiport, 2 or more solids in different directions. So in this question, we have that sodium and glucose are being transported. So it has to be 2 of them, right? So it's going to be therefore symport or antiport. But antiport is in opposite directions. So in this case, the answer should be symport, right? That's going to be the transporter. And the process by which 2 or more molecules are transported in the same direction, that's going to be co-transport. So going back to the question, that's what we got right here, symport and co-transport. Next question, which of these is an example of active transport? And then we have the diffusion of oxygen from a place from high concentration, facilitated diffusion, transport of glucose down its concentration, the transport of sodium from a place of low concentration to high. So let's take a look at the figure. Now we have active transport. And we have that this one goes against its concentration gradient. In other words, from low to high. Right? Because usually it's from high to low. Right? That's in favor of the concentration gradient. But if we go from low to high, that's going to be against its concentration gradient. Therefore, active transportation is going to be against or from low to high. Going back to the question, and that's the answer, right? This one, transport from a place of low concentration to high concentration. Next question, recalling factors that affect diffusion. And we have temperature, presence of enzymes, size of molecules. So let's take a look at the figure. I'm going to give you 5 seconds to think about your answer. You can pause the video if you need. Okay, now that we are in the figure, we have diffusion right here, and factors that affect that are going to be temperature, molecular weight, concentration gradient, membrane surface area, membrane permeability. But as you can see here, there's nothing with respect to proteins, right? Proteins are mainly for active transportation, and proteins are enzymes, and there's no requirement of enzymes at all. So then going back to the question, right here we have presence of answer, right? That's going to be the one that is not. As it says in here, which one of the following does not affect the rate of diffusion? And then it's going to be obviously enzymes. Okay, next question. A red blood cell is placed in a hypertonic solution. This means the concentration of solutes in the solution is blank than the concentration of solutes in the intracellular fluid and will cause the cell to blank. I'm going to stop the video for 5 seconds. You can pause it if you have to, so you can think about your answer. So we have hypertonic solution. Hypertonic refers to the solution, so it's going to be either higher or lower. In this case, it's going to be higher. And then swell or shrink refers to the cell. So then let's take a look at the figure. Okay, so now we are right here in the figure and we have hypertonic solution. So what is it that a hypertonic solution is going to do to a cell? So then what happens with the cell is that the cell is going to lose water. That's why it says in here low water concentration. Therefore, since the cell loses water, then the cells will shrink or will crenate. So therefore, in a hypertonic solution, the cell is going to lose water. Therefore, the cell is going to crenate or is going to shrink. Okay, so then hypertonic, shrink. Let's go back to the question. And right here we have higher and shrink. Next question. Which one of the following organelles sorts, cuts, splices, and packages newly formed proteins into membrane-bound vesicles? And we have a smooth ER, rough ER, Golgi apparatus or Golgi complex, mitochondria, and nucleolus. So let's take a look at the figure. Right here we have Golgi complex. Right, got to do with the proteins and forming vesicles right here, secretory vesicles again. So then Golgi complex should be the answer. Going back to the question, and that's what we got right here, Golgi complex.